our work to close out this webinar. So my name is Coley. I'm an assistant professor at MIT in chemical engineering and electrical engineering and computer science. And what I'll talk about today is our efforts to try to navigate synthetically accessible chemical space. And so as framing, I'll point out that I tend to think about the process of therapeutic discovery and of molecular discovery more broadly as a search problem. It's a search through the massive space of hypothetical small molecule structures, hypothetical macrocycles, hypothetical bifunctionals, and degraders. And really what sort of unifies our approach to the rational design of these structures is the scientific discovery process. It's the way that we think about using information to design new structures, then to make them, and ultimately to test them experimentally. And so through this lens of discovery as a search, we should start to think about applying search algorithms, because we have this toolkit of computational techniques, cheminformatics, artificial intelligence, that helps us make decisions that we need to make as we navigate these design cycles, right? So this is a very general way of displaying what we might do. This is just a fancier version of the design, make, test, analyze cycle. For here, I'm also thinking about the context of setting the goal and of human understanding. Now, in a typical discovery process, of course, we're navigating the cycle, we're iterating through it, and we're trying to propose compounds that have desirable properties, each time hopefully getting more information or getting closer to our target profile. And so the opportunities that we think mostly about in our research group is how we might use AI and other computational techniques to help drive this cycle and make decisions automatically and ultimately test those hypotheses automatically as well. And so there's a few specific areas where this comes into play. So we, of course, use computational models to help design structures, and we use them to build structure property relationships. We use design algorithms to come up with novel molecular structures that weren't, maybe in our training sets, they haven't been tested before. And we also think about synthesis, the role of AI in helping us learn what we can make and how we can make it, because synthesis turns out to be one of the formidable bottlenecks, right, as we navigate these cycles. And so even though this is a short presentation, I'll try to touch on a few of these aspects and give you a sense of our philosophy and our approach to discovery. The technologies I'll talk about are best framed in terms of how we define and navigate chemical spaces, and I'll reuse this figure from a review from Alon Esper-Guzik. And so we can think about the traditional virtual screening paradigm where we have a large list of candidate structures and our goal is to find the best one. Now, to find the best one, we'll take that long list and we'll try to search through it and try to understand the properties that each one might exhibit. And so that could be a high throughput docking campaign, or it could be a high throughput machine learning prediction that we apply to select the best structure. Now, increasingly, folks are excited about the potential of generative AI and other aspects of generative design. These kinds of techniques, which have been around for decades, are of sort of re-emerging interest given the new capabilities enabled by deep learning methods, at least in principle, because these techniques can be creative. They can come up with structures that we don't think of, and that is potentially very promising in finding the regions of chemical space with the kinds of unprecedented properties we might be trying to achieve. Now, for both of these, synthesis comes in and ends up being very relevant to inform how we think about molecular design and navigation. So synthesis is also one of the topics we work on most, because if we think about on the first case, these virtual libraries and virtual screening, the way that we tend to come up with these candidate sets is through these make-on-demand virtual enumerations. It's thinking about taking the building blocks that we have in stock, using transformations we know how to run or think we know how to run, and listing out the many, many, many structures that we believe we can access in a handful of synthetic steps. Now, the role of synthesis for generative design is also interesting to discuss. It's a bit different. Typically, we have these models and algorithms dream up new molecular structures that end up having some motifs that look quite reasonable, like the ones at the top, but other times quite unreasonable, like the ones towards the bottom. And so if you are using a generative AI model to come up with new structures, perhaps it's going to follow chemical valence rules and validity rules but it's not necessarily going to give you molecules that you know how to make. And so we'll revisit this partway through the talk and think about how to correct it. But at a high level, some of the technologies that we think about relates to the use of surrogate models to build structure-function relationships. This is a cornerstone of computer-aided molecular design and of drug discovery in general. We've seen, again, over periods of decades, these efforts to correlate aspects of molecular structure with properties using different representations of that structure, whether we use structural fingerprints or descriptors or lookup tables of functional groups. We've already heard a little bit, right, about the use of graph neural networks and other types of deep learning models. That's really been a trend that came around in 2015, 2016, where now instead of trying to take these fixed representations and correlate them with the properties we care about, we can go back to the structure itself and try to build models that go directly from structure to some numerical representation to the property we care about. And in principle, right, the former approach gave us this information bottleneck. But now if we give models access to the full molecular structure as best we can represent it, it should have all the information that it needs to make a prediction. And so we built various types of these models, and many others in the field have as well. And what we apply them to can be both experimental data as well as simulated data. Right? And this was a very nice example just in the previous presentation of thinking about trying to use machine learning models to recapitulate the same results you might find via docking. Right? And so maybe when we run experiments, the goal is to generalize to new molecules where we don't have any data and we can't run the experiments as easily. But we can also train these machine learning models on simulated data where our goal is mostly just to increase the throughput or decrease the cost. It doesn't necessarily mean the accuracy is better. At the very least, we're getting improvements in cost. And so that actual use case is one that we'll start with now in thinking about the role of these surrogate models in structure-based drug design. So a couple years back, we were sort of looking at the increasing rate of high throughput docking screens being published, reports of hundreds of millions or billions of candidate ligands screened against a single protein of interest. And through these really massive campaigns, interesting hits were discovered and interesting series were then advanced. But the computational investments to dock, hundreds of millions or billions of compounds is substantial, right? Docking is cheap, but it's not that cheap. And so in this work, we simply took a framework of model-guided optimization and tried to apply it to this use case. So rather than trying to exhaustively screen 100 million candidates where we know most of them are going to be sort of underperforming, we would evaluate some of them. So we dock a subset, we train a surrogate model, and we use that model to steer us in the direction of promising chemicals. Tell us what parts of chemical space to avoid and which ones to favor. We would iteratively select which molecules were worth docking. And so when applying this, what we can find is that we get a significant enrichment, right, compared to a simple random screen as you would do in a brute force search, where after only 2.5% of the calculations, we can already find about 90% of the best molecules, where here best is just defined in terms of raw docking score. And so this kind of workflow is not making docking more accurate, but it's letting us leverage our existing computational infrastructure and use it to screen a larger space. So we're still using docking as the source of truth for this study, but by inserting this model in the framework, we are allowing ourselves to search a larger space with a smaller fraction of the actual experiments. And so we have this computational savings of about 40x, 40-fold. Now, even though sort of here we're thinking about drug design, sometimes people I talk to don't. And so this notion of model-guided optimization is, of course, very general, and it can be applied in any number of situations. And so in this other use case, looking at optimizations of molecular properties derived from TD-DFT calculations, which are much more expensive than docking, several hours instead of seconds, we can get the same acceleration and the same enrichment. And so this kind of surrogate modeling and optimization, again, is really a cornerstone of computer-aided molecular design, what we do. Now, we also so now turn our attention to generative design, because I think this is expanding the toolkit that folks are excited about in this area. And we too think about applications of generative models to different targets and different sort of therapeutic goals. So this animation is just showing the results of generating a PROTAC against a simple surrogate model trained on PROTAC-DB, so a proxy for degradation performance. And you can see it's stitching together what looks like a fairly plausible heterobifunctional. So this isn't experimentally validated. This is not claiming this is an actually good PROTAC, but the model is learning to optimize this property oracle that we're giving it access to. And so these generative models do this kind of atom by atom or fragment by fragment generation. That's quite common. But the failure mode of these techniques is something that I already sort of indicated, which is that we can get structures proposed by these models that sometimes look reasonable and sometimes look very unreasonable. And one of the ways in which they look unreasonable is that they'll have these structures which are not easily synthetically accessible. And so we can't really take these into the lab or send them to a CRO to make. Just doesn't make sense. And so there's some simple heuristics we can build in to try to account for this, but really the problem is synthesis. We really want these models to recommend actionable suggestions for our chemists to test. And this notion of sort of synthetic accessibility getting in the way of generative design is something that's ubiquitous in the field. And so we can go all the way back to this 2019 paper, which now feels like a very long time ago, from in silico medicine, where they used a generative technique to propose 30,000 structures. Different sorts of cheminformatics techniques narrowed that down to 40. But from those 40, they still only were able to make 6. And that determination of narrowing down to 6 was based on synthetic accessibility. And so we have this pipeline where in principle, machine learning is responsible for some aspect of design. But after proposing 30,000 structures, you only make 6 of them, right? There's a lot still of intervention that happens in this process. And this exemplifies, I think, why we worry about synthesis, because it does serve this, it narrows down what we're able to test, right? It's a pretty big bottleneck for us. And so when we're making new molecules, right, we have a designed molecule on a whiteboard or on a computer, that necessitates designing a new synthetic route. So we can sort of go back and remind ourselves, okay, what is a synthetic route? What is the information that we need to share? And so for this example of palbociclib, which is a known molecule with known routes, we can sort of see that the slide denotes the recipe, right? We have a roadmap that tells us how to connect from purchasable starting materials through intermediates all the way up to our final product after a series of different synthetic steps. And so we think about techniques related to computer-aided synthesis planning, which also involves the use of algorithms to do this planning process for us. And so at a simple level, we can teach computers to do retrosynthetic analysis. This has existed since the 1950s. There have been approaches for computer-aided synthesis design for decades and decades. What was sort of pivoted in more recent years, maybe we'll call the last decade, is that there's been an increased interest in using reaction databases to propose the routes. And so by reaction databases, I'm thinking about resources like SciFinder and Reaxys, the USPTO, Pistachio. These give us examples of chemical reactions that were run experimentally, who ran them, what reactants they put in, what conditions they used, and what major products they either found or were expecting, and what the yield was. Not all resources have all this information, but this is, in a general sense, what we have access to. We're missing some of the details of the synthetic process that we would like to use for learning, but nevertheless, we have access to reactants, products, and some notion of conditions. And so these examples really form the backbone then of a whole class of data-driven synthesis planning programs. So we can train models to learn the strategy of what reactions are possible based on the literature. There are way too many techniques to summarize in just this one talk. Suffice to say that there are many different algorithms that have been developed over the years to do this task of retrosynthetic analysis from data. So dozens and dozens just in the last couple of years. So among them are techniques that try to learn transformation patterns based on reaction templates encoded as smart strings. There's also language-based models that try to use transformers and other architectures to go between One smiles of a product and smiles of the reactant structures. There's also graph translations, because again, graphs, as we've seen, are a very intuitive way of representing molecular structures, atoms being nodes, bonds being edges. And then some other architectures, including graph to smiles, which we published last year, which tries to combine the best of both worlds. Right, so we try to take a graph molecular encoder and combine it with a sequence or a language decoder to learn this translation from products to reactants. So again, there are many different techniques, far too many to give them justice here. But for any number of these one-step retrosynthetic models trained on the literature, we can start applying them recursively to the targets we want to make. And so we can apply them to a product that's been proposed either by a chemist or by a generative model, and we can get recommendations of how to make them in one step. Because we can't purchase those starting materials, we go further back and further back and further back. And so there are complexities here that I'm skimming over related to how one does this multi-step search, how one manages the complexity and the combinatorial space of options as you do this recursive expansion. But with some well-established search strategies, it's not that complex now to have the capabilities of proposing these multi-step synthetic routes that connect a product of interest to commercially available starting materials. In our case, a lot of the capabilities that we've been developing over the years get rolled into a program that we call ASKCOS. So ASKCOS is really a suite of these different synthesis planning modules. It's sort of grown organically over the past maybe 5 years or so, thanks to funding from DARPA, the MLPDS Consortium, as well as the National Institutes of Health. And ASKCOS really tries to bring together different machine learning models and cheminformatics tools that are broadly useful for synthesis planning. So that includes retrosynthesis, but it also includes other tasks of recommending reaction conditions, predicting the products of reactions, and maybe even the impurities of reactions, as well as tools like classification, atom mapping, and sort of selectivity prediction. And so, we have some out-of-date statistics, but ASKCOS is really where we've brought together a lot of our predictive chemistry models to try to make them accessible to the chemistry community. And so, we can use ASKCOS to evaluate and filter the proposals from generative models, but we're also thinking longer term about how to combine synthesis and molecular design. This is not a strictly new idea, right? Folks have been trying to combine the idea of reaction-based enumeration with generative design for some time. And really the idea here is that if we want a model to propose structures that we know how to make, let's not ask it to propose structures atom by atom and bond by bond, but let's ask it to propose structures building block by building block and reaction by reaction. And so in this particular work, we train a neural agent, a policy network, to select commercially available building blocks, select reaction transformations defined by templates that we can encode. And this agent is going to learn to stitch together a synthetic pathway from the bottom up. So we give it access to maybe 100,000 building blocks, maybe 100 reaction types, and it's going to learn how to compose a tree. The synthetic tree is a synthetic route. It corresponds to a particular product of interest. And so, end to end, what this looks like is a model that takes some conditional code, some embedding of the target, and proposes a new product molecule. But in doing so, it's had to propose a synthetic route, and so that means we know how to make the molecule. And so if we sort of abstract away all of the insides of this, we can then think about wrapping this whole setup with a genetic algorithm. And so we can say, well, instead of making this particular molecule, what if I adjust that conditional code and change what it is I'm trying to make? Well, if I change this conditional code on which the whole generation process is based, we end up getting a different synthetic tree, which means we get a different molecule and then different properties of that molecule. And so we can use a genetic algorithm to then optimize properties of the molecular structure that is made, all the while guaranteeing that every structure that is proposed is associated with the synthetic routes. And so as sort of a highlight of what that really gets you, in effect, when we use our model to optimize this heuristic score against GSK3 beta, it's a simple machine learning model doing a binary classification. We end up with a structure that is extraordinarily simple and is accessible in one synthetic step, as opposed to if we use fragment-based approaches, we tend to get sort of very large, bulky structures that don't make much sense to a synthetic chemist, even if they score highly according to the model. And so it's this sort of regularization that is really beneficial here for us. Again, just as sort of another example, sort of to highlight this point, if we learn to compose a molecule by learning to select building blocks and reaction types and stitch them together into intermediates, and then learn when we're done and when we have a final proposal, we end up being able to do all the same things that we do for other de novo design algorithms. Like we can use docking as the oracle. We can try to find a new molecule that has a better docking score than some known ligand. We can do all that same stuff while guaranteeing that every molecule that's proposed is associated with the synthetic routes. Now, there might be selectivity concerns, there might be quirks of the routes as a result of the templates that we have defined, but this model is trying to align our use of AI to propose new structures with the reality that when we have those new structures, at some point we're going to have to make the molecule. So this is sort of what I mean when I talk about learning to navigate synthetically accessible chemical space, right? We're trying to bring together molecular design and synthesis planning to be sort of one integrated process. So again, we come back to the general notion of this discovery cycle. We think broadly about how we can bring AI and different sorts of computational tools in to address different aspects of the decision-making process and the decisions that one has to make when navigating this cycle. There's a few of the particular areas that I highlighted in this brief talk that we work on. So that does include the modeling side, so building different sorts of models that understand how to correlate structure to function. It's the design side as well, so coming up with either new molecular structures or trying to select the best molecular structures from a very long list of candidates, maybe hundreds of millions or billions. And then the synthesis side, where we worry about what reactions are possible. And we do this all so that we can drive these design cycles more quickly. And so, really just a couple of final takeaways here. AI models for property prediction is a cornerstone of computer-aided molecular design. It's useful in both contexts, both sort of ways of finding molecules. When we're trying to design new structures, we have to be a little bit more careful about what we can actually synthesize and what we can make in the lab. And there are ways of doing this, right? We can build these models that integrate understanding of chemical synthesis of feasible reactions with the goals of molecular optimization and try to bring them together and come up with ideas that can be actionable, whether we send it to a CRO, we synthesize them in-house, or ultimately we synthesize them with a robotic platform. So I'll close there, just acknowledge funding support from various organizations and groups over the years. I think in particular, I'll highlight the Machine Learning for Pharmaceutical Discovery and Synthesis Consortium. I'll acknowledge the great group members that I get to work with who have actually carried forward all of this research, and thank data sources of CAS, Pistachio, and Reaxys. And I'll be glad to spend the next couple minutes answering any questions, although I know this is the end of a long day. So thanks very much.